Welcome to AP Biology 1.2 Elements of Life. In this chapter, we will look at the elements that make life and specifically look at why six of those elements carbon, hydrogen, nitrogen, oxygen, phosphorus, and sulfur are uniquely suited for creating biological molecules. We'll also look at the properties of carbon and we'll look at the four macromolecules, which are proteins, carbohydrates, lipids, and nucleic acids, and look at the elements, the elemental composition of those four macromolecules. And then we look at the geometric isomers and enantiomers, and finally end with a spotlight on functional groups. There are 118 elements in the periodic table. 90 of these occur naturally. It's important to recognize that while we do not understand the nature of the force called life, we know that the physical structure that supports life is made of elements that have been recycled across time. And so the same elements that make stars is what make us. So it's kind of a fascinating thing to think about. So even though there are 19 naturally occurring elements, 6 of those elements, carbon, hydrogen, oxygen, nitrogen, phosphorus, and sulfur, dominate our body mass. So they make up 96% of the body mass. And the question is, why? It has to do with the kind of chemical bonds that are formed. So if you remember in our last chapter, we looked at 3 major chemical bonds. We looked at covalent bonds, which involves sharing of electrons, and we saw that there were polar and non-polar covalent bonds, depending on whether the electrons are shared equally or not. Then we had ionic bonds, where there is an exchange of electrons from one element to another element, and the resulting ions are attached to each other using electrostatic forces, which we call as ionic bonds. And then we looked at metallic bonds, which are present in where atoms are arranged in a crystal-like structure with a sea of electrons between them. Now, the types of bonds that are formed define the nature of the molecules that they create. So, for example, the stability of the molecules that are being created depend on whether the bond is ionic or covalent. And also whether the products that are created are stable or can have certain amount of flexibility to be converted back to the original reactants. So in that context, ionic bonds are unstable in the sense that when you have products form by the breakdown of an ionic molecule, it is very difficult. It's very stable to the point you cannot bring it back to its original composition without too much of energy expenditure. So ionic bonds are unstable in that sense. It's difficult to bring back. And so in biology, we need a certain balance between reactivity and stability. If it's too stable or if it is unreactive, then it is not going to work, and ionic bonds are unstable. Metallic bonds, on the other hand, form crystal-like structures. So because of the way they are arranged in a lattice-like form, there is a limitation on how they can be branched out. And otherwise, life will look like a whole bunch of Lego blocks, so which is not desirable. So covalent bonds are the choice for life, because not only do the way they are able to form bonds allow them to make very complex structures, they are able to be they are stable enough and they are reactive enough so that those bonds can be broken and reorganized. So if we look at that as a requirement for creating biological molecules, you can see which of these elements in the periodic table are suitable for forming covalent bonds. And if you look at this list here, of these two groups, you have the alkali and the alkaline earth metals, you can immediately eliminate those because they can only form ionic bonds. So they are not going to be suitable for life. And the groups from here all the way to here, they are elements that form metallic bonds. Again, given that metallic bonds form crystal-like structures, we can eliminate them. You can also eliminate the very last group. This last group is stable. They are the inert gases. So they have no reactivity, if at all. If at all they have it, it's very little. So they are not suitable for forming chemical bonds. So you can leave that out. So what that leaves us is this group from 4A going down here to 7A. The problem with 7A is halogens. That includes chlorine and fluorine and bromine and iodine. And these are quite reactive, quite explosive in their reactions. So they are not suitable for biological life. So that leaves us only with this little group of elements. Even in this little group, you have elements that are too big to form stable bonds. So starting from germanium all the way to polonium, you see that these elements can form chemical bonds, but they are too large to hold on to the chemical bonds that they have formed. And this has biological implications. So think of arsenic. Arsenic has very similar properties to phosphorus, and because of the number of valence electrons that it has, it's very similar to phosphorus. It is the same as phosphorus. However, arsenic is much larger than phosphorus. So let's say in a biological molecule, arsenic replaces phosphorus. And after the bond is formed, arsenic does not have the power, the electronegativity, to hold on to the electrons. So the bonds that are formed are unstable, and therefore they can break apart quite easily. So for example, look at DNA. DNA has a backbone structure. We will look at the structure of DNA in detail later, but the backbone of DNA has phosphorus, phosphate sugar backbone. And that phosphate sugar backbone, imagine if that phosphate is replaced by arsenic, the bonds will form, the backbone will form, but it will break apart very quickly, and therefore it will kill the organism. And that's the reason why arsenic acts as a poison. When you introduce it into a biological system, it can replace phosphorus, but after it forms the bonds, it will break apart very quickly. So because of the large size, you can eliminate all these elements starting from here to here. Okay, so this block is also gone. What that leaves us is 6 elements here that are able to form covalent bonds: carbon, nitrogen, oxygen, silicon, phosphorus, sulfur, and then of course you have hydrogen sitting there on the top. Now, of these 6 elements, carbon is the only one that can form bonds, chemical bonds in 1D and 2D structures. So one-dimensional and two-dimensional structures. So carbon becomes a much more interesting candidate for creating complex biological structures compared to the other candidates here. So while all of these hydrogen, carbon, nitrogen, oxygen, phosphorus, and sulfur are important for life, carbon becomes the most important molecule because it not only meets all the requirements for balance between reactivity and stability, but it can form the most complex structures. Silicon is similar to carbon, but it is not a good candidate for life, and we will see why in a minute. Carbon has 6 electrons, and 4 of those electrons are valence electrons. As we saw in the previous chapter, per the octet rule, it needs to have 4 more electrons in the valence shell to satisfy the octet rule. And this can be done by sharing electrons with itself or with other elements. And the bonds that can form, there can be a single pair, there can be 4 single pairs of electrons being shared, or there can be double bonds, meaning that 2 pairs of electrons can be shared with another element, or it can be triple bonds with 3 pairs of electrons shared with another element, and so on. So that again gives a whole lot of variability in how you can form complex structures. Also, so again, it can form a variation of, it can have 4 single bonds, it can have 2 single bonds and a double bond, or it can have a triple bond and 1 single bond, and so on, right? So it has all of these combinations. Also, carbon has a low electronegativity. Just to recap, electronegativity is the ability to attract electrons. So carbon has an electronegativity of 2.6. Hydrogen has an electronegativity of 2.2. So it's very close to hydrogen in terms of its electronegative capabilities. Compare that to oxygen, for example. Oxygen has an electronegativity of 3.8. So that low or the moderate electronegativity makes it a suitable element in terms of the bond formations that can happen with carbon. So as we saw, carbon can form long chain compounds with itself, and that's called catenation, or it can form compounds with many other elements. It's very versatile. So the question then comes up is, why not Si? Why not silicon? So it's interesting because silicon is much more predominant in our Earth's crust than carbon. Silicon is 28% of our Earth's crust, and carbon is only 0.02% of our Earth's crust. And still carbon is preferred over silicon for life. Why is that? There are several reasons. One is that silicon has very, very strong interactions with water. And so it is unstable in the water, along with water, in the sense that it cannot be reconverted. And the second one is that it bonds preferentially with oxygen. So it's a unidirectional reaction with oxygen, and it's going to steal oxygen from other elements and not going to let it go. The third thing is that we, as we have evolved right now, breathe carbon dioxide. And carbon dioxide, CO2, right, is in the form of a gas. Silicon dioxide, on the other hand, is a solid, and it's otherwise called as sand. So that again is not amenable for life as we have experienced it. So here's a really interesting video from PBS that talks about why silicon is not a preferred element for life, and I would welcome you to take a look at that. So now we have established why some elements are more suitable for life than others. So looking at how elements are assembled, you have water, of course, which is important for life, and small molecules like ADP and ATP. And then you have very, very complex molecules like proteins, really large complex proteins, and really large complex carbohydrates and lipids, and a very long chain of DNA and RNA. And also these can interact with each other. Proteins and carbohydrates can interact with each other, and carbohydrates and lipids can work with each other, and so on. So there is a high complexity of arrangement of molecules within systems. There are 36 trillion cells in the human body, and each cell has about 100 trillion atoms. And you can imagine the complexity of choices that come up, and how each of these have to be carefully coordinated. And this coordination happens because of the innate chemical properties of the elements that form biological molecules. And once you have the small biological molecules, they organize into macromolecules. And there are four major biomacromolecules: carbohydrates, lipids, proteins, and nucleic acids. We will study them in detail in the next three chapters, 1.3 to 1.5, actually 1.3 to 1.6. We will study the four biological macromolecules in more detail. But what we want to look at here is what is the composition, the elemental composition of these four macromolecules. Carbohydrates have carbon, hydrogen, and oxygen, and they are in the ratio 1:2:1. So think of glucose, for example, C6, which means 6 carbons, H12, 12 hydrogens, O6, 6 oxygens. So that's in the ratio of 1:2:1. Carbohydrates always have that ratio. Lipids also have carbon, hydrogen, and oxygen, and they may also contain nitrogen, phosphorus, and sulfur. But as far as carbon, hydrogen, and oxygen go, the ratio is different from carbohydrates. It's 1:2 of carbon and hydrogen, and then oxygen is less than 1 always. So there's less oxygen present. Proteins have carbon, hydrogen, oxygen, nitrogen, and sometimes sulfur. Nucleic acids have carbon, hydrogen, oxygen, nitrogen, and phosphorus. Okay. So I just wanted to point out, for example, an amino acid here. An amino acid has carbons forming interactions with itself, and carbon forming interactions with oxygen, oxygen forming interactions with hydrogen, carbon forming interactions with hydrogen, carbon forming interactions with nitrogen, nitrogen forming interactions with hydrogen, and so on. So all of these are arranged in a particular way and form that makes it that makes things work. But it's not just the sequence of arrangement of atoms, but it is also how they are arranged, the sequence in which they are arranged, and also the spatial arrangement of atoms. So this is where isomers become important. Isomers are molecules with the same molecular formula, but they have differing arrangement of atoms. So if I have 6 carbons, 12 hydrogens, and 6 oxygen, 6 carbon, 6 oxygen, 12 hydrogen, there are multiple ways to arrange that. So they would have the same molecular formula, but depending on how I'm arranging it, I could end up with different molecules. Those are isomers. Stereoisomers are a class of isomers. They have the same sequence or arrangement of atoms, but they have a different arrangement in three-dimensional space. And one of the classes of stereoisomers is geometric isomers. The order of atom bonding is the same, but spatially you have a difference. So, for example, this molecule here is called dichloroethene. You have 2 carbon molecules. Each of those carbon is attached to this white stuff, that is the hydrogen. And then you have the green circles, that is chlorine. So in this arrangement, you have the 2 chlorines pointing in the same direction. If it is in the same direction, this geometric isomer is called as a cis isomer. And if you have the chlorine pointing in the opposite directions, that makes it into a trans isomer of dichloroethene. So this cis versus trans becomes really important when we study lipids, because that is going to change the properties of the molecules, the lipid, the fatty acid tails that are being created. Again, we'll study this when we study lipids, but cis versus trans, something to remember. And then we have optical isomers. So we call these enantiomers. Here we have a picture of lactic acid. We have L-lactic acid, levolactic acid, and dextrolactic acid, D-lactic acid. So what you're seeing here is a representation of lactic acid as mirror images of each other. So if you take your hand and place it in front of you, and you'll see that your two hands are exactly alike, but you cannot superimpose them. Right? If you place your palms on top of your or on top of each other, you'll see that your thumbs are pointing out in the opposite direction. Right? So that non-superimposability is what makes optical isomers. So the lactic acid, L-lactic acid, and the D-lactic acid are mirror images of each other. So why are we looking at, why is all this important? In biology, structure is function. So when we think of a biological molecule and what makes its properties, we have to look at the composition of the biological molecule. What are the elements that make it, and also the arrangement of the atoms in the biological molecules, what sequence they are, but also how they are spatially arranged, as in the case of cis versus trans isomers, which will give unique properties for the biological molecule that it's part of. So, speaking of function, one of the major things that we need to remember in our AP course are functional groups. And this is one of those few places where you might have to memorize structures and names in biology. So a functional group is a group of atoms within a molecule that give a unique characteristic to that molecule. Like, for example, O and H, okay, oxygen and hydrogen together. That's the hydroxyl group, OH. So this is a group of atoms. Oxygen and hydrogen are a group of atoms. And together, this group, whenever it is attached, whatever molecule it is attached to, it will give that molecule a unique characteristic. It will make it hydrophilic, and we will call it as an alcohol. Okay, hydrophilic, as you know, is the ability to work with water, right? And hydrophobic is inability to work with water because it is non-polar. So I can put this OH on a molecule called methane, CH3, and it will become methanol. I could take that OH and add it on a slightly larger molecule called ethane, and then it will become ethanol. So it is transferring a specific characteristic to whichever molecule it is attached to. That's why it's called a functional group, because it transfers a function. A group of molecules transfers a function to a particular function to whichever molecule it is attached to. Also, if you look at molecules, functional groups are the phase of interaction, meaning that that's where all the molecular, intermolecular interactions between molecules happen, right? Interactions between molecules happen. So there are a few biologically significant functional groups that we need to remember. The first one is aldehyde. So if aldehyde is going to have a central carbon with a double bonded oxygen, and then another carbon on one side, which is attached to other things. We call the group here that is attached to carbon as R, as a variable group. It could be anything. It could be CH3, it could be CH2, something attached to something. So we call it as a variable group. But the other side of the carbon is attached to hydrogen. So CHO, that's an aldehyde. Aldehyde is a little bit different from ketone, because ketone, instead of having hydrogen on one side, it's going to have, again, a double bonded oxygen, but also the carbon is attached to two variable groups, two carbon atoms attached to something else. Okay, So carbohydrates have aldehydes and ketones. Then we have a group called amino group. Amino group is NH2, and the N is attached to a variable group, to a carbon. So NH2, it has the ability to attract H plus ions. So if you remember when we were studying pH, we said that what makes a solution acidic or basic is how much of H plus ions are present in it. If there is a lot of H plus, then that solution is acidic. If there is very little H plus, then we will have a basic or an alkaline solution. So NH2 has the ability to attract H plus molecules, and because of that, it is reducing the H plus concentration in the solution. Therefore, it is basic. Right? Amino groups are basic. Carboxylic acid or carboxylic groups have C, O, and the C is attached to an O, which is attached to an H. So it's COOH. And carboxylic groups have the tendency to lose the hydrogens. What that means is they are increasing the H plus concentration in the solution. So that makes them basic. So we'd call these as carboxylic acids. If you take an amino acid, for example, this is an amino acid, and as you know, amino acids are the building blocks of proteins. They are the monomers that make up proteins. And all amino acids have the basic structure of a foundational structure where you have a carbon, which is attached to a hydrogen on one side, and an amino group on one side, and a carboxylic group on one side, and a variable group on the other side. So an amino acid has a carboxylic group as well as a basic, an acidic group as well as a basic group. That's why it's an amino acid. It's called amino as well as an acidic group. Then we have esters. Esters are carbon double bonded to O, and you have a variable group on one side. Then you have oxygen, which is attached to another variable group. So COO, that's an ester group. Another important group here is phosphate. So phosphate groups have a phosphorus molecule attached to 4 oxygens, and that can be replaced variably, right? So one of those can be replaced with a variable group, and so phosphate groups are really important in DNA and RNA because they form the sugar backbone structures. Also, phosphates, the phosphate group is important in ATP and ADP, the energy currencies of our cell, because energy is stored in the bonds of phosphorus and oxygen, which when broken down is going to release energy. And then in amino acids, we have 2 amino acids, cysteine and methionine, that have a group called the sulfhydryl group, S and H, sulfur, hydrogen, so sulfhydryl group, and attached to a variable group. So sulfhydryl groups can form bonds between different amino acids. And those kinds of bonds are called as disulfide bonds. And that's again an important structural characteristic for proteins. So all of these aldehydes, amino groups, carbonyl, carboxylic, ester, hydroxyl, ketone, phosphate, sulfhydryl, all of these are polar groups, polar functional groups. And quickly recap, polar is unequal sharing of electrons because one of the elements has a higher electronegativity compared to the other element that it's sharing electrons with. So for example, in water we saw that oxygen has a much higher electronegativity compared to hydrogen. Therefore, the electrons are attracted closer towards itself compared to hydrogen, which gives oxygen a slightly negative charge and the hydrogens a slightly positive charge. So in all of these molecules, you have a polar situation wherein oxygen is is pulling the electrons closer to itself than the carbon that it is attached to. Or in the hydroxyl group, again, with oxygen attracting the electrons closer towards itself compared to the hydrogen, and so on. So remember that hydrophilic molecules work well with, are those molecules that can work well with water, and basically because those molecules are polar or ionic. Okay, so all of these functional groups have very good water interactivity. The one exception to this, among this, or one functional group that is not hydrophilic but hydrophobic, and that we will study today, is the methyl group. So the methyl group, it's simply carbon and 3 hydrogens attached to it, and one side you have the R group, the variable group, which could be another carbon, and if it's for example another CH2 molecule, then you will have ethane, right? So what we have here is carbon and hydrogen. When they share electrons, their electronegativities are very similar. So like I said, carbon electronegativity is around 2.6 and hydrogen is 2.4. So the difference in electronegativity between those two is only 0.4. So if the electronegativity difference is less than 0.5, what that means is they do not have, neither of these elements have the ability to pull the electrons towards each other preferentially. So their electrons are equally shared, which creates non-polar molecules. So methyl groups are non-polar functional groups. And there's one other bond that functional group that we want to look at, that is the amide functional group. And the amide functional group is formed, it's really important in proteins. It's created when you have an amino group of one amino functional group of one amino acid interacting with the carboxyl functional group of another amino acid, and you have a loss of water. Okay, and that loss of water results in a bond between this carbon and this nitrogen being created, and this carbon, of course, being attached to its oxygen and another carbon and so on. And here you have this carbon, this nitrogen being attached to a carbon and so on. So this carbon and nitrogen are forming a bond. That kind of a bond is called as an amide bond, otherwise called as a peptide bond. So again, we will look at this kind of synthesis of amino acids, the synthesis of proteins from amino acids in the next few chapters. So here are the things that we want to recap and remember. We need to remember why the balance between reactivity and stability and the ability to form complex structures in three-dimensional space makes carbon and oxygen, nitrogen, phosphorus, sulfur, and hydrogen the most suitable elements for life. And we looked at carbon as the singular element because of its versatility in forming different kinds of bonds, and because of its moderate electronegativity and its ability to catenate, which is to form links of molecules with itself, and also its ability to form to form covalent bonds with a bunch of other elements, makes it a much more suitable molecule for life. Um, and of course, that silicon is not a suitable molecule because of multiple reasons, because of how it forms a unidirectional reaction with oxygen, and how strongly it interacts with water. And we also saw that molecules have to be arranged are arranged in increasing level of complexity, and that allows for um, life to function um, effectively. And then we saw the molecular composition of the four major macromolecules, and also we saw that in addition to just the composition of the molecules, it is the sequence in which they are arranged, and also the way they are arranged spatially, that make a difference in the functionality of a biological molecule. Finally, we looked at the functional groups that are important for life, that are going to be repeating motifs that will give characteristics of biomolecules. So hopefully this was helpful. In the next 3 chapters, we are going to look at the 4 macromolecules, and in the sixth and in the next one, the 1.6, we are going to spotlight on nucleic acids. Okay, hopefully this was helpful. Thank you.